in pathology and has been working on epigenomics and genomics of myoplastic or myelodysplastic syndromes, MDS, and AML, leukemia. And so today she's going to be talking about epigenomic approaches, provide novel biological and clinical insights for myeloid malignancies.
Thank you, Maureen.
Thank you.
Good morning, everyone. Today I'm going to talk about how to use epigenomic approaches to provide us novel biological and clinical insights for myeloid malignancies. Epigenomic studies have been widely used in cancer biology, and it helps us to uncover the disease heterogeneity and novel subtypes, and provides us insights into the mechanism of the disease and propose the mechanism for therapeutic response and help us improve the translational applications. So in this talk, I'm going to use myelodysplastic syndromes as an example to illustrate how we address these three aspects of questions. So MDS is one of the most frequent hematological malignancies, especially in the elderly population. This disease is characterized by the dysplastic changes in the bone marrow with increased bone marrow cellularity and ineffective hematopoiesis. And MDS patients have high risk of progressing to acute myeloid leukemia. Based on the disease aggressiveness, MDS patients can be classified into 4 categories by the International Prognostic Scoring System, and as shown in this table. I'm sorry, wrong button. And the low and intermediate MDS patients have relatively higher survival rate, and these patients are considered low risk MDS, and they occupy above 70% in all the MDS patients. And in the first study, I'm going to talk about these low-risk MDS patients. So there are some clinic challenges for MDS prognosis. In clinic, we know that for these low-risk MDS patients, some of them will stay at a relatively stable stages, and we think this part of the patients is stable MDS, which means there is no change in their lab tests, and the frequency of the supportive treatment for these patients the same, doesn't change within at least 18 months. However, there are some of these low-risk patients can quickly progress to aggressive stages, which means they have to have increased transfusion rate, and the number of blasts in the bone marrow is significantly increased within 18 months. But in clinic, currently there is no way to distinguish these 2 types of patients due to the limited knowledge of this disease. And these patients were treated equally just by some supportive transfusion treatment. However, for those progressive patients, we know they need more aggressive treatment to prevent their progression. Previous epigenomic studies on MDS have proved that there is some aberrant DNA methylation in these patients, especially for those more aggressive patients. However, it's not clear if this abnormality is present in the early stage or it's just a consequence of the disease progression. So we are asking, are there specific epigenetic features at diagnosis that can help us to predict the disease progression stages for these low-risk MDS patients? So to answer this question, we select some low-risk MDS patients samples based on their known clinical records and collect their primary samples from the Memorial Sloan Kettering Cancer Center. Specifically, we selected 13 stable MDS and 7 progressive MDS, and we have all the medical records for them for at least 18 months. And average age would be around 50 or 60 years old. Yeah. Mm-hmm. The end of the what? The yes, we also collect. Yeah, for the progressive patients, the sample collect at the time of their progression. And then we also collect their diagnostic samples at time of the diagnosis for this same set of patients. So they're pairs. So totally we have 20 baseline samples and 20 matched follow-up samples. We performed bisulfite sequencing on these 40 samples, trying to figure out if there is any methylation difference between these patients. Yeah. PBMC. Oh, okay. Yeah. Because yeah, actually ideally we get the bone marrow samples. We actually get them. Actually, we collect totally 60 or over 60 samples, and but within those bone marrow samples, the quality is pretty low. We cannot use them. So that's our second choice. So we focused on these peripheral blood mononuclear cells because they give us a decent number of cases in each stages. So it's a trade-off. Yeah. Okay. So the method we use is called enhanced reduced representation bisulfite sequencing, ERBS, which is basically an enhanced version of the traditional RBS method. So this flowchart on the left-hand side highlight those major steps in this protocol. So by using this method, we can use as low as like 2.5 milligram genomic DNA as the input materials. And then they applied the restriction enzyme called MIST1 to digest this DNA to get some smaller fragments, which will help increase the bisulfite conversion rate and increase the library quality. And then they did end repair and methylated adapter ligation. And another step, they modified is they introduced the size selection step in this protocol, which help us to find more enriched CPG fragments and increase the CPG coverage across the genome. And then they did the bisulfite treatment overnight on this sample, followed by PCR amplification and then sequencing. So using this method, we can get like 3 million CPG sites, including those distal intergenic regions. Compared with the traditional RBS, which only focused on the CPG islands or promoter regions, but lack of the representation for the distal intergenic regions. This protocol significantly increased the CPG coverage on the genome. And we can get a bisulfite conversion rate as high as 99.8%, actually above this score. And the pipeline, analysis pipeline we used contains these, the general steps, we contain these steps. So we first trim the adapters and then align the sequence to the bisulfite converted reference genome and did differential methylated region analysis followed by some gene annotation and downstream functional analysis. So by applying this method, we check our data and did the differential methylated region analysis. We found that at baseline level, there is a distinct methylation profile exists between progressive and stable MDS. So showing this volcano plot, those 350 TMRs highlighted by these red dots, and also can see their methylation level in each of these samples for these DMRs. Because most of them are hypomethylated DMRs in progressive patients. And when we check the methylation levels at the time of follow-up, we found these distinct methylation profiles become more pronounced. And the number of DMRs almost doubled than the baseline. And again, most of them are hypomethylated in the progressive patients. And interestingly, we found the stable patients at follow-up is more consistent with the normal peripheral blood samples. So we did prove that these methylation difference exists at the time of diagnosis. Maybe in the future we can develop some biomarker from this data to help us distinguish these two stages of patients. Mm-hmm. Yeah, it's not a big number actually. It's relative. But we do. Yeah. Yeah. No, no. This is just put the normal samples. Yeah. But and also we apply biological cutoff with absolute methylation above 25%. It's quite a stringent cutoff for the methylation difference. Okay. What? Yeah, I also apply. Yes, I also apply FDR 0.1 because the method the methodic is quite stringent. Yeah. Okay. So and then we annotate these DMRs to the genomic regions and some regulatory regions. We found that for both baseline and follow-up DMRs, they tend to be depleted from the CDG sites, promoter regions, while enriched at the more distal intergenetic regions. Is that for these hypermethylated DMRs at follow-up samples, they kind of enriched in the CDG, CDG island. But remember, these hyper DMRs only occupy a very small number of, a very small proportion of the total DMRs. And also, we found both of these DMRs are enriched in the enhancer regions, which indicate their regulatory role in the genome. And next, we checked if there was any overlap between the baseline and the follow-up DMRs. However, we didn't find very strong overlap. Yeah, sure, sure. Mm-hmm. But you see the distribution. Yeah, yeah. Yeah. Mm-hmm. Okay, so as I mentioned, there is a little overlap between these two sets of DMRs. And we are surprised by these results. Actually, we're expecting with the disease progression, we could add more DMRs, but it should be based on the previous difference, but it's not. And before I talk about that overlap, we did the gene annotation for these DMRs and did some gene set enrichment analysis. This is just two representative enriched pathways that these DMR associated genes enriched in. Here I listed the Wnt signaling and the cell cycle pathways. And we know these pathways are well involved in the cancer development. So it makes sense. And as I said, the observation is not like our expectation, like there should be more overlap between these two sets of DMRs. But when we check the methylation levels for the baseline and follow-up samples at those union set of those DMRs, we do find these methylation levels were quite correlated, which means although the DMRs identified in the follow-up samples are not exactly same as the baseline DMRs, but the direction of the methylation is quite consistent with the baseline. So here, the blue dots are the union set of DMRs in the stable MDS, and red dots are the DMRs in the progressive MDS. As you can see, the overall correlation is pretty good, and then even for the stable MDS, it's even better. And we also observed that the stable MDS at these DMRs tend to be hypermethylated, as you can see the density for these cells at this corner. And we also observed the dispersion for this methylation level in progressed MDS is quite higher. Yes, we think there are some methylation variance existing in the progressive patient, and that might introduce the difference of the methylation set. And then I checked the methylation variance only on those follow-up MDS across the 4 type of patients. And we can see for the methylation variance in the progressive patients is significantly higher than baseline, than the stable patients, even at the baseline. So we think there are some variance, some methylation variance exists in the progressive MDS, and that might help us explain the shuffling of the DMRs in our data. And at the same time, we found a paper recently published on genome biology. In that paper, they studied epigenetic complexity and the difference of that complexity between 2 stages. We think this method may help us to explain the underlying mechanism of this methylation variance. And on the other hand, the ERBS data we have provide us sufficient coverage per CBD site, which allows us to apply this method to further study the methylation variance in our data. So briefly, this method mainly talks about the clonal epigenetic shift, and the whole story based on the concept of epi-allele, which is the short term of epigenetic alleles, referring a specific DNA methylation pattern of genetic locus. As an example shown in this cartoon, so this big circle represents a bulk of cells, and it contains 12 different individual cells represented by this white circle. And within the particular genetic regions, and in each cell, there are 2 copies of methylation pattern, for the adjacent 4 CPG sites. So those methylated sites were represented by these black dots and white one unmethylated. So if we look at the combination of the 4 adjacent CPG sites, we are expecting 16 different combinations. So which means we have 16 different epi alleles in this situation. And then they calculate their complexity or the combination of different epi alleles in the first stage and compare it to the second stage. They can have a score to evaluate these epi allele shifts. So you can see there are some difference in the epi allele composition between these two stages. And in our case, we're gonna compare the shift between the stable MDS and the progressive MDS. So in ERBS data, each rate can be considered as a representative samples for epi allele. So in this case, we have totally 22 epi alleles in each stage. And by applying some entropy theory, we can calculate the difference between the epigenetic composition in these two stages. And if this difference is significant, then we consider this loci have the epigenetic shift. And we call this loci as E-loci. So basically, we require these rays spanning at least 4 adjacent CPGs and with the coverage greater than 60. And then this is the workflow of this method called NetClone. And basically, the input is the alignment SPAM file, and they collect the epi allele composition, calculate the frequencies and the entropy between different stages, and output all the alleles with significant shifts. And each E-loci is defined at a particular entropy cutoff. So by applying this method, we checked our E-loci in between the stable and progressive patients for both baseline follow-up, and then also checked the overlap with our DMRs. We see that at follow-up, we are seeing less epigenetic shift than the baseline, and these overall these E-loci have very little overlap with DMRs. So basically, this method is looking at different genomic loci than our DMR. So it cannot directly answer our question if the methylation variance is due to the epigenetic shifting. But I also think maybe our tiling strategy may influence the overlap, because we require the DMRs to be non-overlapped 25 base pair windows. But these E-loci are focused on those regions with the rates exactly spanning 4 CPGs and have coverage greater than 60. So this may introduce some artifacts. But anyway, since we have this data, we can check if these significant shift alleles are have some regulatory roles. And again, we annotate them to the genome and some regulatory regions. We found that they also depleted from promoter and the CPG island regions, but enriched in the enhancer regions. So, and then we, because we have all the combination of the epi alleles in each stages, we can easily calculate their overall epigenetic polymorphism, which is give us idea of the overall complexity of the epi allele patterns. So we calculate polymorphism score for both progressive and baseline samples, and calculate their difference. We make this plot, and we found that this epipolymorphism difference is significantly higher in the follow-up patients, which indicate that there is a high epigenetic heterogeneity changes for the follow-up MDS cases at those eloci. And also, we found out that based on the epigenomics difference, we can sort of observe two types of epiallele shift models, showing by these two example locus. So in the first locus, we observe that there is the epiallele become more homogeneous in the progressive patients, indicated by the decrease of the epipolymer score in the progressive patients. And we call this model a selection model. However, in the second loci, we can see the heterogeneity increase in progressive MDS, indicated by the increase in the epipolymorphism in progressive patients. And this model is called drift model. And when we look at the distribution of these two types of models, for baseline and follow-up samples, we observe that in the follow-up samples, the drift model increased a lot than the baseline. So again, this data indicates the follow-up eloci have increased epithelial heterogeneity in the progressive MDS patients. So by this type of analysis, we sort of get a sense that there was some local methylation pattern difference exists in the progressive MDS, especially in the follow-up. And this may help us to explain why there were such different DMR exist between baseline and follow-up. So to summarize the first part of the story, we identify the distinct methylation profiles between progressive and stable MDS patients, and these distinct profiles already exist at diagnosis. And the DMR-associated genes are enriched in cell cycle and Wnt signaling, which are closely related to cancer development. And we also found the increase in epiallele heterogeneity for progressive MDS at follow-up, which might lead to the shuffling of the follow-up DMRs. So next, we are interested to see the current therapies and their response for the MDS patients. Unfortunately, there is no curative therapy other than a bone marrow transplant for these patients. And bone marrow transplant is a really viable therapy because different factors. And there are also two types of DNA methyltransferase inhibitors called azacitidine and decitabine have been widely used to treat these patients. And these two drugs are basically the cytosine analogs. It acts by incorporating to DNA synthesis and trigger the DNA damage. However, there is only about 40% of the patients can respond to these drugs. And we have to wait up to 6 months to see if these patients can respond or not, which means for those non-responders, we are kind of wasting their time to see the response. So it is very urgent to develop method to predict their response at the time of diagnosis, such that we can pick up other therapy for those non-responders. So currently in clinic, we cannot do that. So we're asking, are there specific genetic or epigenetic features at diagnosis can help us predict the clinical response to these drugs? To answer this question, we collect some samples from a clinical trial in Italy. And in this trial, they look at a subtype of MDS patients called chronic myelomonocytic leukemia, CMML. It has overlap characteristics with MDS patients, but with myeloproliferation feature. So totally, we have 40 CMML patients. They were all treated by decitabine for 6 months, and we have their response results evaluated by the clinician. So we collect their bone marrow mononuclear cells at the time of diagnosis, and then perform ERBS RNA-seq to trying to figure out if there is epigenetic and transcriptional features exist between responders and non-responders. So far, there is no publication have shown any DNA methylation profile difference between responders and non-responders, because those studies are all focused on promoter-centric methylation data, like P450 array data. So they cannot find a difference. But in our data, we did find some distinct DNA methylation profiles between responder and non-responders at the time of diagnosis. So again, we find the 160 EMRs, and the half of them are hypermethylated. And when we annotate these DMRs to the genome, we did observe that these DMRs were depleted from promoter regions and CBG island. And this proved our hypothesis that that's because of the limitation of previous methods, made them fail to identify this distinct profile. However, with the genome-wide method, we did find this difference. And these regions enriched in distal intergenic regions and enriched at enhancers. And then next we asked, since there are some distinct methylation profiles between responders and non-responders, can we utilize this data to build epigenetic classifiers to make the prediction? So we use the spot vector machine as our classifier, and using the cohort we have to train the model, did some feature selection that is embedded in 10 cross validation, and found some features and train the model. And then we apply this model to an independent cohort to validate our findings. So specifically, by using the first cohort, we identified 21 methylation sites as well our feature in the SVM model. And by clustering using these 21 features, we almost separate these two groups of patients. And then we train the SVM model using the 21 features and the first cohort, and make it prediction in the second cohort. We found the model is pretty robust when predicting independent data. So, but there is a trade-off, because the sequencing data is a stochastic process, so we cannot guarantee those features we identify in the first cohort can all appear in the second cohort. So if so in this data, we found only 6 of the out of the 21 features can all appear in the 28 samples in the second cohort. So if we just use these 6 features, we can get the accuracy only like 71%. But if we increase the number of features we used, and by sacrificing the number of predicting cases, but still we have enough sample to predict, the accuracy increased up to 87%. So in the future, we're gonna design some like sort of cheap array to test only the 21 features we selected and trying to develop that as a biomarker for clinical application. Sure. Okay, so I combined the gene filtering and kind of greedy forward feature selection procedure. So first of all, I look at all the whole genome CBG size, and we have the supervised analysis comparison between progressive and stable patients. We have some raw p-value. I use that p-value to filter out those non-significant ones. Just keep those ones that have potential distinct between this group. And then I rank these features by evaluate the performance of each of these filtered CPG sites and have a ranking. And then I did forward feature selection by including each one of the features into the model and then test their performance to find the best set of CPG sites. Oh yes, because yes, because only 15 samples can all contain these 16 features. Yeah, yeah. Because the coverage, the second set of our cohort doesn't cover the same set of CDG in the genome because it's stochastic matter we cannot control. Yeah, ER based. Yeah, it's not targeted. It's reduced representation. Oh yeah, another thing I need to mention. Yeah, so the first cohort is the 40 samples I just mentioned when we checked the methylation difference. And these samples are the unsorted mononuclear cells from bone marrow. But the second cohort is from another clinical trial. It's the same patient treated by the same drug with same strategy, but these samples were sorted monocytes. So it's more clear population. Not same patients, totally different. It's an independent clinical trial. And then but still, even we use the unsorted cells data to train the model, we still can get robust results when predicting those sorted cells by using this method. No, actually they're tiled. Yeah. I'm sorry. Oh, the kernel I use the Gaussian, I think. And then we asked if there is a transcriptional program associated with this drug response. And so we did some RNA analysis, RNA-seq analysis, and then we did find some different differential genes between these two group patients. And here shows the gene set enrichment test. We found those differential genes are more enriched in DNA replication and cell cycle, which makes sense because this drug acts by acting incorporating DNA synthesis at the S phase. So we think there are some mechanism in some genes involving cell cycle or DNA replication may be the underlying mechanisms for the drug resistance. And we check the profile for those differential and methylated genes, and we found that there are only a small proportion of upregulated genes for those non-responders. And out of these genes, we found two genes very interesting called CXCL4 and 7. These two genes are chemokines, and they have been reported to involve in the cytotoxic effects in the DNA synthesis process at S phase. So we think these genes might be a good candidate to fish out the mechanism of this drug resistance. And by the histoimmunochemistry assay, we proved that these two genes are overexpressed in non-responders at the protein level. And we also proved that these two genes can block the DAC-induced cell death in the primary CMML cells. So this is the cell viability assay. So if you look at those control, which means those cells were only treated by decitabine without CXCL4 and 7, the cell viability is significantly decreased, which means the drug effect well. But if we add the CXCL4 and 7, this reduction of variability was totally relieved. And we also proved that these two genes can abrogate the effect of the DAC on the normal CD34 cells. So those two genes might be the good candidate for the novel therapy for these patients. However, we currently there was no direct evidence showing what's the effect of this drug on these patients. So people don't know if these effects were due to the cytotoxic effect or some epigenetic change or genetic change caused this response. So in another study, our collaborator investigated the methylation levels before and after these drug treatments. And we found a lot of DMRs were hypomethylated after DAC treatment. As shown by this chromosome distribution for the DMRs, those green ones are all the hypomethylated DMRs, and the magenta ones are the hyper ones. So you can globally the reverse of hypolation in the patients after the DAC treatment. However, these are in the responders. However, in non-responders, there's only few DMRs, and the number of hyper and hypomethylation are almost equal. And again, if we look at the global methylation level, we see the non-responders shows similar methylation levels as untreated patients, while in responders, the methylation level overall decreased. So in that study, our collaborators also ruled out the possibility that the genetic burden changed by this drug. And so they think the major effect of this drug on these patients is the reversal of the hypermethylation. So this is the first time we've shown that this drug acts on the patients by reverse the whole methylation genome. So this is kind of a little bit about the drug mechanism. And to summarize this part of work, we identified the methylation-based classifiers and prove that have high accuracy when predicting the response to the drug at the time of diagnosis. And by gene expression profiles, we found that cell cycle programs is involved in the response to this drug, and found two genes overexpressing non-responders and block the DAX effect on primary cells. So these two genes can be our candidate target for alternative therapy. And we also proved that the response to these hypomethylating agents is associated with a global reversal hypomethylation. And this is pretty much for my talk today. And I also list some of my other works. So recently we have a publication on the cancer discovery. We proved the methylation patterns in single mutation of these DNMT3 genes with FLT3 ITD mutation is comparable to acute molecular, acute myeloid leukemia, and these mutations, single mutation, actually promote the aggression of the mouse model to develop to AML very quickly. So another two projects I'm working on in my PhD study is about using the ontology data and literature data develop gene network and use that network to predict the biological signaling or the pathway modulated genes. And finally, I'd like to share some future directions of my current work with you guys. So we're looking at the role of hydroxymethylation in drug resistance for MDS patients, because we know that ERBS data cannot distinguish methylated CDG site from those hydroxymethylated CDG sites. And we know hydroxymethylation level is more correlated to gene expression. So we are using sort of immunoprecipitation method to check the hydroxymethylation signals in these patients. And by integrate with the ERBS data, we can distinguish these two signals and can see how these signals play the role in drug resistance. And also because the gene annotation for DMRs is open question, so we don't know if the annotation is accurate enough. So we need some 4C sequencing data to help us validate our annotation strategy. And this is also a going on project. Okay, I'd like to thank my mentor, Maria Figueroa, who provided me a lot of opportunity to work on different projects and get a lot of experience about epigenomics and cancer biology studies. I really appreciate her mentoring. And I also thank all my lab members, especially the two previous members, Christian and Jason, who did all the library prep and bench validation work for the study I just talked. And I also thank our close collaborator, Maureen, who gave us a lot of advices and help on our data analysis. And I also thank her to give me this opportunity present here. And I also thank Alex, who involved in all the ontology fingerprint work. And then other collaborators from Italy, France, who gave us all those precious primary samples. Without their support, we cannot make the story. And I thank my former mentor, Jin Jen, and Dr. Lu, who helped me a lot for my PhD studies. Okay, I'd like to take any questions. Yeah. Because for those data, we need you're talking about time cost, right? We have difficulty to get the exact time when we collect the samples. So, which means the clinical information is not ideally complete for those patients. We only know they're around 1.5 years old, so we don't know the exact time numbers for those follow-up patients. So that's one thing, and I did the pairwise comparison, and actually I didn't find differential methylated regions between those patients. Yes, yes, I compare that. Yeah, which one? Oh, the first. I think he's talking about the first study of the paired samples. Oh, that's the in the third study. Yeah, that's when you check the mechanism of the methylation. Yeah, they are paired. But we don't have the method to take care of the paired analysis for methylation data right now. So we we don't have the method to evaluate that paired difference. So we treat them as an independent group. Oh, that I checked the overall correlation. They're pretty good for that set of data, for the the final set of the treatment, before treatment, after treatment, for individual patients. The overall methylation is the anti-correlated. Mm-hmm. You mean using one paired sample to identify the signature? Um, I have some. Is that? Oh yeah, in PCA analysis, for that piece of data, I didn't have the data here, but um for the first study, I did the unsupervised analysis, and like this. Um, I this line just connects the baseline, the different groups of samples, and the circle just show these samples project majorly in this area. So that's a good question actually. I didn't pay much attention to that because we focused on the DMR analysis, and currently there is no paired method allowed to increase the power, and so we just treat them as individual in a group. Mm-hmm. Yeah, yeah, yeah. Majority of the because we also apply the tiling strategy. We that's not the individual CPG size. Maybe the CPG size are covered by both cohorts, but when we tiling these samples, we consider the coverage for each individual samples and make decision how to telling them for the starting point and endpoint and make the exclusive window. But yeah, I do see a lot of difference when you make the tile uh, between the two cohorts. Oh, that's for epithelial analysis. Yeah. Yeah, yeah. But here the coverage is the same as from 10 to 500 as the default we use. Yeah. Yes. Yes. Yeah. I think they are symmetric. There are some, we checked some genetic mutation panels in these patients, but we didn't see the correlation between the responders or the different two stages of patients. I didn't show the data here. And these, I have it in supplementary. For example, so this is the mutagenetic, well-known somatic mutations in MDS, and we checked their status in these patients at different stages. Because these are low-risk MDS patients, the genomic mutation burden is relatively low overall in these patients. And so that's why you look at this figure is quite sparse. So we only have few mutation events within the samples. But overall, we didn't find the significant correlation. And this is first one. And second one, about the response, we also check the somatic mutation for these commonly mutated genes between responders and non-responders, and there was no significant disease. And these mutations are not correlated with methylation level either. So we think the epigenetic effect will be the explanation for it. Yes, exactly. We need to have more samples further evaluate. Yeah. 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 Exactly. Yes. Yeah. We didn't check. I this is not. Yeah. We didn't check that. Yeah. Yeah. Correlation. As far as I know, we don't know that. Yeah. So yeah, ideally we should use bone marrow sample, but in this case, the cohort is bad. Yeah, it's totally different. Yeah, I know. Yeah. We always have some problem with this cohort for this study, and yeah, actually I started the project since I joined the lab, but it took so long for me. protein together because we have a lot of problems in the middle of the cohort. Yeah. Yeah. And the first project actually designed also by some trying to finding biomarkers, but we end up with very few numbers of cases. It's not possible to do that. So we just report this difference we observe. Just we cannot make a solid conclusion from that study. Yeah. We only have like 14 samples for RNA-seq data, so that's impossible because of the quality of the RNA. Yeah. Yes, we really hope we can. Quality is degraded a lot. Yeah, we only can have 14 samples. Yeah, it's difficult to deal with this. Yeah. Thank you for your attention. Thank you.